So my research is in biomedical photonics, and that means that we use light to interact with biological matter, and that gives us information about several important processes. For example, I'm doing a lot of research related to skin. So here in the biomedical photonics lab, we have an experimental multiphoton setup. So it's a femtosecond laser. So it's near infrared light, so it's invisible to our eyes. But the thing is, they are high energy pulses and very short pulses of femtoseconds. And when these pulses come to the sample, we can get fluorescence. So it's a laser scanning technique, so it's an optical table and you have the laser light coming here, and we have scan mirrors putting the light into the microscope where we then can place the sample and scan. In most of the cases we use tissue here. So the practical use is that we can apply these techniques to study, for example, pharmaceutical aspects like topical drug delivery, how new drugs can penetrate into the skin. We can also study toxicological effects like sunscreens. It's a lot of debate now whether they are good or bad. And these effects, we could also apply this technique for studying. And of course, we can also apply this technique to study skin dysplasia and skin cancer, and potentially be also a diagnostic tool. We have an experimental setup here in this lab, which is very then complementary to the more hands-on tools that we have at the Sahlgrenska faculty, where we also have a clinical instrument which can actually be used directly on patients, and we also have a core facility where we can take biological samples straight and do microscopy directly on those samples. So we have sort of the whole spectrum, all the way from molecule to patient.
We have been collaborating in many years with the Department of Chemistry and Molecular Biology, trying to implement optical advanced microscopy techniques in medical research. What this machine does is that it takes images of a skin disorder or a skin cancer in order for us to make a diagnosis. It is a non-invasive technique. You just apply the microscope on the skin, and the microscope shows images of cells, and then you can, from these images, make a diagnosis. It doesn't cause any pain or discomfort for the patient, and it doesn't cost so much because it's only the images that you have to produce in order to make a diagnosis. In the future, we are very positive that this technique can allow us to actually set all the diagnoses in skin, both the inflammatory diseases like psoriasis and eczema, of course, all the skin cancers also.
Yeah, so the future challenges are really related to developing more contrast and to be able to pinpoint specific cells that are dysplastic, so they are abnormal. Because the problem is that we get a lot of information from these type of techniques, a lot of images and a lot of gigabyte of data that has to be interpreted. So image interpretation combined with contrast mediators, which can sort of highlight the things that we're looking for. And here we are specifically studying how we can use nanoparticles as a technique, as a biosensing tool, so that we can really see the things that we're after in a biological tissue.